lecture, we will study about the introduction of enzymes. Today we will talk about the introduction of enzymes. The chemical reaction of enzyme, how enzymes work, the process of enzyme, the enzyme structure, and how do enzyme work. The lock and key hypothesis about enzyme and the induced fit hypothesis about enzyme. So let's get started. First of all, what is enzyme? Enzymes are proteins. Enzymes are macromolecule biological catalysts which they speed up biological chemical reaction. Molecules upon which enzyme may act are called substrate, and the enzyme convert the molecule substrate into different molecule known as product. The enzyme glycosidase converts the sugar maltose to 2-glucose sugar active site reduced in the red, maltose substrate in black. Structure of enzyme. Enzymes are generally globular proteins. Structure determines the function of enzyme. The reaction takes place in a small part of the enzyme called active site, while the rest of the protein acts as a framework. The amino acid around the active site attach to the substrate molecules and hold its position while the reaction takes place. This makes the enzyme specific for one reaction, only other molecules would not fit to the active site. Enzyme activity initially increases with temperature until the enzyme structure unfolds denaturing, leading to optimal rate of reaction. Now, how do enzymes work? The first hypothesis is the lock and key hypothesis. Emil Fischer in 1890 suggested that each enzyme had a particular shape into which the substrate fit exactly. This was known as the lock and key hypothesis. According to this hypothesis, substrate is imagined like lock and enzyme is imagined like key. A specific key can open a specific lock. Similarly, specific enzyme can break only a specific substrate, each enzyme having active site and substrate, so the specific enzyme can break only a specific substrate. The active sites of the enzymes are a rigid structure that does not modify or change during the reaction process. However, later on, the induced fit hypothesis Daniel Koechlin modified to do the lock and key model of Pasteur in 1958. Enzymes change shapes by substrate binding to form enzyme-substrate complex. For example, hexokinase has a large induced fit motion that flows over the substrate adenosine triphosphate and xylose substrate in like. For something interesting, join us in a Facebook group link in the description to know more about everything and something. And finally, thanks for watching.